As technology improves, new techniques are developed to analyze and reanalyze fossils of long-extinct animals to help bring them to life beyond anyone's wildest dreams. One of those technological advancements is the visualization of microscopic parts of the feathers and skin of ancient animals that helps to piece together the true colors they once carried. At this point, there is a quickly growing list of extinct animals for which the colors and patterns are being sussed out. Obviously, such an analysis requires the most pristine fossils that can be found, so that list will always be small, no matter what. But we are now at a time in which that list can't be counted on a single hand. One of the landmark cases of color in extinct animals is that of Anchiornis. The small, feathered holotype fossil of what would become known as Anchiornis was first dug up in the Yaolegao area of Jiangcheng County, Liaoning, China, in the early 2000s. The holotype of the critter was first described by Zhu Xing and colleagues in 2009 in their paper in the Chinese Science Bulletin. They named the animal Anchiornis huxleyi, with Anchi for early or near and ornis for bird, due to the animal's evolutionary relationships, and the species name in honor of Thomas Henry Huxley, who was kind of the first guy to vehemently argue for the dino-to-bird connection. The holotype of Anchiornis is an articulated skeleton missing the noggin, a chunk of the tail, and the right forelimb. Since this first paper, over 255 specimens of the animal have been recovered and reported, with many more in private hands and plenty of this massive cache remaining to be described. The little critter's remains are so numerous that virtually every aspect of its anatomy, inside and out, are known. At a glance, the animal was quite similar to Archaeopteryx, but also plenty of other bird-line theropod dinosaurs that were essentially awfully close cousins to true birds. But the closer you get to them, the blurrier and grayer the lines become, so let's not touch on that. It's because of those blurring lines that Anchiornis has been classified as a member of the Avialae, near to Archaeopteryx, but also as a troodontid as part of the more dromaeosaur line non-bird dinosaurs, as a member of the Archaeopterygidae, and most recently as part of its own group, the Anchiornithidae, as an early offshoot of the Paravian group before the troodontids, dromaeosaurids, and birds. So all of that kind of just means it's a weird birdy, dinosaur-y bird thing. It plays a major part in the evolution of birds and bird-like non-birds, but like 100% what it is outside of being its own offshoot group is a work in progress. Aside from the soft tissues preserved in many of these specimens, and aside from the feathers preserved in many of these specimens, the next most important thing, and perhaps the number one most important thing depending on your perspective, are the colors. What the hell kind of colors were smattered across the feathers of these little dino birds? Anchiornis was not the very first dinosaur to have its coloration described by scientists based on physical evidence. That award goes to Sinosauropteryx, a small compsognathid theropod also from China. Paleontologist Nicholas Longrich was the first to bring the idea of Sinosauropteryx's color to the Society of Vertebrate Paleontology in 2002. Before him, the team of researchers that described the little dinosaur's remains in 98 or 99 had thought that the obvious alternating light and dark banding of the feathers in the fossil were an artifact of preservation, or just how the rock cracked when the fossil was excavated, as it is a part and counterpart. A 2010 study would prove Longrich correct, as well as be one of two landmark studies that showed the process for visualizing the microscopic cells that once held color in ancient fossils. This study, by Fusheng Zhang, Stuart Kearns, Patrick Orr, Michael Benton, Zhanghe Zhao, Diane Johnson, Xing Xu, and Xiaolin Wang, looked at a few dinosaurs. An isolated feather, the carcass of the enantiornithine bird Confuciusornis, Sinosauropteryx, Sinornithosaurus, and even made sure to compare them to feathers of some living dinosaurs. The technique they used, quite simply, involves using scanning electron microscopy and just scanning a bunch of fossils. Obviously, there's a whole lot more to that than I know, but we don't really need to know it to understand how it works. The fossils are so perfectly preserved that microscopic structures have been preserved in the soft tissues and feathers. Specifically, the spaces that once held melanosomes are preserved. A melanosome is an organelle found in animal cells and is the site for synthesis, storage, and transport of melanin, the most common light-absorbing pigments found in the animal kingdom. Melanosomes are responsible for color and photoprotection in animal cells and tissues. So, all you have to do is sample some spots on a perfectly preserved fossil that seems to be a good candidate for melanin preservation, send it through the SEM, and then fuss about the results to interpret what they mean. Such a task was undertaken by another team of scientists at virtually the same time, but this was with a new specimen of the then recently described Anchiornis. The specimen, EMNHC PH828, comprises part and counterpart of a partial skeleton in 3 shale blocks, with elements of the forelimbs and hind limbs in near complete articulation. Preparation was minimal and most feathers are well preserved, even to their insertions in the skeleton or soft tissue. Elongate, pennaceous forelimb and hindlimb feathers are present, as are contour feathers associated with the skull and body. The team, which consisted of Chongguo Li, Keichin Gao, Jacob Vinther, Matthew Shockey, Julia Clark, Liliana Dalba, Xingzheng Meng, Derek Briggs, and Richard Prum, sampled parts of the feathers from far away from the body or distally, and close to the body or proximally, and from all body regions. They sent 29 samples into the scanning electron microscope, which revealed impressions of spherical to oblate carbonaceous bodies, or impressions 100 to 1908 nanometers in length, which they identified as melanosomes. Most samples revealed elongate eumelanosomes that varied slightly in morphology and distribution in different regions of the body. This specimen of Anchiornis was the first to have a relatively complete noggin, preserved with a triangular crest of feathers on the back of it. Those feathers contained distinct subspherical phaeomelanosomes, about 500 nanometers in length. A sample from the back and underside of the skull showed distinct regions of spherical and elongate melanosomes. There are different types of melanin, by the way. The different types usually correspond to different shades and colors, but are chemically different variations of the same chemical. There is eumelanin, which comes in 2 types, brown and black. There is also phaeomelanin, which often results in a range of colors from yellowish to reddish. This is all good and everything, but the team, or any team analyzing these things, needs to be absolutely sure that the shapes they are looking at correspond to the correct shape of melanosome that produces a given color in a living analog, like living bird feathers, so that's what they did. For comparison, the team assembled a dataset on melanosomes from a phylogenetically diverse sample of living bird feathers, with black, gray, and brown melanin pigmentation, but lacking structural coloration. A reminder that structural coloration is responsible for many blues and virtually all iridescent and metallic colorings in feathers, exoskeletons, hair, and skin, and is just a rearrangement of layers, pigments, and air bubbles. Other molecular pigments, such as carotenoids and porphyrins, also produce plumage colors but are not preserved. Thus, the team couldn't address their possible effects in this Anchiornis. 4 properties of melanosome morphology and distribution, long axis variation, short axis skew, aspect ratio, and density, were used as variables in a mathematical variable analysis they ran on the living bird melanosomes. In general, eumelanosomes that produce black and gray colors are long and narrow, whereas those that produce rufous red and brown colors are short and wide. They assigned colors to the fossil samples on the basis of the statistical analysis of modern feather colors. All the melanosome morphologies in the fossil fell within the range of those observed in living birds. 24 samples were assigned a color with more than 0.9 probability. 3 samples were ambiguous, with probabilities between 0.56 and 0.74, and 3 samples lacked significant numbers of melanosomes and were interpreted as unpigmented. Sample 14 was a distinct outlier in the analysis, probably because of its high aspect ratio and long axis variation, and was predicted as gray. Melanosome shapes usually strongly correlate to the pigment they hold, but not so when it comes to gray. Gray can show up due to a whole bunch of variations on melanosomes. Samples from the body contour feathers, samples 10 to 13, indicated a black or gray color. Samples from the marginal forelimb covert feathers, samples 1 and 5, and the front and top surface of the legs, samples 14 to 16 and 18, were also black or gray. The undercovert feathers in the propatagial region were predicted as brown, but with low probability. Samples from the tips of the elongate primary and secondary feathers of the forelimb, samples 4 and 6 to 8, the toes, sample 19, and the elongate pennaceous feathers of the hind surface of the shank and foot, samples 20 to 24, were reconstructed as black. Melanosomes in sample 17 are poorly preserved, preventing the interpretation of color. A heterogeneous sample from the contour feathers on the side of the face, sample 27, showed separate regions of distinct melanosomes and was reconstructed as containing distinct, adjacent, black and rufous feathers, or parts of feathers. Samples from the long forecrowned feathers and the shorter feathers from the sides of the crest, samples 28 and 26, indicate gray, whereas the longest feathers from the center of the crest, sample 25, indicate a brown in close proximity to rufous. The discriminant analysis clustered sample 25 with rufous feathers of living birds. Melanosomes were sparse in bottom portions of the elongate pennaceous feathers of the forelimb and the hind limb, samples 2, 9, and 15, indicating that these areas were lightly pigmented and whitish in color. The correlation between the colors reconstructed on the basis of melanosome morphology and density and the preserved appearance of the fossil supports extrapolation of at least dark and light color patterning. For example, part and counterpart of the Anchiornis specimen exhibit dark tips on many of the upper covert feathers of the forelimb and the hind limb that are consistent with eumelanin pigmentation. Put together, these results, result in an Anchiornis that looks something like this. Anchiornis was darkly colored with gray and black body plumage. The head was gray and mottled with rufous and black. Elongate gray feathers on the front and sides of the crest appear to frame a longer rufous hind crown. Gray marginal wing coverts formed a dark epaulette that contrasted strongly with the black and gray spangled light primaries, secondaries, and greater coverts of the forelimb. The large black spangles of the primaries and secondaries created a dark outline to the trailing edge of the forelimb. The large black spangles of the primaries and secondaries created a dark outline to the trailing edge of the forelimb plumage. The spangles of the outermost primaries were black. The greater coverts of the upper wing were spangled with gray or black, creating an array or rows of conspicuous dots. The contour feathers of the legs were gray on the shank and black on the foot. Like the forelimb, the elongate feathers of the backside of the hind limb were white at their bases, with broad black distal spangles. Unfortunately, the tail is unknown in this specimen. However, this is an hypothesis. The pigments and the areas they correspond to are accurate, but their exact placement in a greater body pattern is variable because samples weren't and couldn't have been collected from literally every single surface of the specimen. This is why every single piece of paleoart you will see of Anchiornis is slightly different, and should be. There still remains some variability in how one can reconstruct this specific specimen. To add to that level of uncertainty, this study was done on one specimen of one species of Anchiornis. Colors and patterns could have and likely did differ between sexes, ages, species, regional populations, subspecies, and more. If you're going to recreate this animal, shake it up a little bit. I'm sure you've noticed that the plumage color pattern elements of Anchiornis are strikingly similar to various living birds, including domesticated fowl. The identification of melanin-based pigment within and among feather plumage coloration patterns in Anchiornis helps reveal the evolution of color pigmentation patterning mechanisms in dinosaurs. For example, the pattern observed in the simple tail integumentary structures of Sinosauropteryx has been interpreted as representing spaced light and dark patches among feathers. The Anchiornis results confirmed that melanin-based patterning is a mechanism for within and among feather variation, producing lighter and darker regions. According to the data from Anchiornis and its phylogenetic placement, these two common mechanisms of plumage patterning in crown group birds are supported as minimally having a first appearance in the most recent common ancestor of Troodontidae and crown aves. Complex within and among feather plumage coloration, such as that in Anchiornis, is used in display and communication in living birds. Such communication, however, may function in different ways, commonly in intersexual communication, and less so in interspecific and intraspecific competition for restricted foraging. For example, multiple species of antbirds forage on army ant swarms. Alternatively, bold plumage color patterns can function in interspecific threat and defense postures, such as in some owls or the sun bittern. In startling predators or warning conspecifics within a flock, or in startling invertebrate prey that are seized as they attempt to flee, such as with North American Setophaga redstarts. Neotropical Myorioborus whitestarts, and Australian Rhipidura fantails. Melanin deposition in the distal portion of primaries in birds, such as gulls, may offer added resistance to wear. The plumage of Anchiornis is composed of a variety of differently sized pennaceous feathers that vary little in feather shape. The body contour feathers, the primary and secondary forelimb feathers, and the forelimb and hindlimb coverts all share a similar aspect ratio. Thus, mechanisms producing varieties of feather shape may postdate the evolutionary first appearance of plumage color variation. The most recent common ancestor of Anchiornis and birds must have had the capacity to develop white, gray, black, and rufous plumage color patterns both within and among feathers. Both mechanisms of melanin-based plumage patterning evolved before the derived form of active powered flight. The observed color pattern in Sinosauropteryx is consistent with only among feather variation in melanin pigment deposition. Such variation occurs in birds and would be optimized as ancestral to at least the Coelurosauria group. However, the evolutionary origin of within-feather pigmentation patterning, including stripes, feather spots, and spangles, appears later. Its origin is presently coincident with the origin of elongate pennaceous feather structure in the most recent common ancestor of Maniraptora, when data from the early Oviraptorosaur, Caudipteryx, are considered. Thus, the first evidence for plumage color patterns in a feathered non-avian dinosaur suggests that selection for signaling function may be important in the early evolution of feathers. That's pretty much it for the colors of this little critter. Well, until another study was published on another specimen of Anchiornis in 2015, but that's a story for another time. For more interesting stories about nature, the history of life, or what goes bump in the night, subscribe, like this video, drop a comment in the comment section below, and hit the bell icon to stay in the know with everything Edge. Thanks for watching.
Mm-hmm.